Welcome to this week's work in progress. I'm really excited to introduce Dr. Patrick Pearson from NEWVEC this week. Dr. Pearson is a postdoctoral research associate for NEWVEC and the microbiology department at UMass Amherst. He is studying ticks and tick-borne pathogens, and his current work focuses on the biology of Borrelia burgdorferi, deer-targeted tick control, and routine surveillance of deer and their ectoparasites. So, Pat, I will let you take it from here. Thank you.
Great. Thank you, Jane. So hi, everyone. Thanks for coming to this work in progress. Like Jane said, I'm Pat, and today I'll be talking about Project Whitetail. So Steve, or Dr. Rich, actually is the person that coined this term for this project. And really what this is an umbrella term describing all the whitetail deer related research that's going on in the lab. So I'm hoping today I can give everyone just an update and an overview of where these projects stand. But I'm going to be talking about 6 projects today, and as the name implies, Project Whitetail, deer are at the center of all of these projects. I think this is a really good venue or forum to talk about all these different projects because they are all works in progress. And deer are at the center of these. And really, since I want to talk about 6 different projects, I'm just going to skip over the top of most of these. I'm not going to go into too much detail on any one of them, but I do want to get everyone up to speed of where they are and at least introduce the topic to everybody. The first project I want to talk about is host-targeted tick control, or more specifically, deer-targeted tick control, which I think at least some people in the audience have heard about before and have an idea of what that is. And then I want to talk about a project using deer blood to study the impact of deer blood on Borrelia burgdorferi survival. And then I want to talk about how we can use deer to survey or do surveillance of ectoparasites and their pathogens, how we can use deer to investigate understudied ectoparasites, so things like deer keds, how we can use deer to study their parasites, the parasites that live and infect deer, and then how we can use deer to do a serosurvey of arboviruses. And I want to point out that these first 2 projects are possible or coming out of a new research station that we developed, which I'll get into in a second. And these last 4 projects are all possible based on samples that we're collecting from a hunter-harvested whitetail deer surveillance program. So I'll start this way, and then I'll just work around these 6 projects. So I'll start with host-targeted tick control. So before I really get into it, I want to just give everyone a quick overview of tick life cycle and the sort of rationale behind doing host-targeted tick control, or more specifically in this case, deer-targeted tick control. So what I'm showing here is the life cycle of Ixodes scapularis, or the black-legged tick. So Ixodes scapularis is a medically important tick in the Northeast United States, and it's the vector for a lot of common pathogens like Borrelia burgdorferi, which is the agent of Lyme disease. And what we think is that the adult stage of the tick, of Ixodes scapularis, feeds primarily on white-tailed deer. So the males and females will both get on white-tailed deer. They'll meet each other and mate. And if it's a female, she'll take a final blood meal, which will give her the energy to lay eggs, and the tick life cycle will continue. So we think of deer as reproductive hosts for this tick species, and really important for maintaining tick populations. And so the idea behind deer-targeted tick control is if we can inhibit or limit this interaction right here between adult female ticks feeding on white-tailed deer. And the idea is, if we can limit that feeding, there will hopefully be fewer ticks because the tick life cycle will crash. Our tick populations will go down. So hopefully there'll be fewer ticks. Hopefully there'll be fewer infected human biting ticks. And then that hopefully result in less incidence of tick-borne disease in people. So that's the idea behind deer-targeted tick control. But really, to test different methods to keep ticks from feeding on white-tailed deer, you really need, or it's helpful at least, to have a controlled environment to thoroughly test these deer-targeted methods. So that's what we set out to do recently here at NEWVEC. we wanted to build a facility to do these sorts of experiments. And we did just recently, last summer, late summer of 2024, we built this facility, which we're calling the Cervid Vector Ecology Research Station, or CVERS for short. So this is new, at least in the last year. Again, it was built in the summer of 2024. It's on UMass forested property, pretty close to campus, and it's about a 5-acre fenced enclosure in the woods. And we have capacity, we think, for about up to 15 white-tailed deer. And the layout is pretty simple. It's essentially a large rectangular shape in the woods. It's enclosed by this 8-foot fencing. There's a couple things going on inside the station. So there's this buffer area where we just use primarily for storage, but it's also another layer of security between the outside world and the animals. And then we have this smaller enclosure where we, the research team, interact mostly with the animals, and that's where all the things are that are necessary for husbandry, like food, water, and shelter, and things like that. So we built the facility, and then of course we had to populate it with animals. So we got our first cohort of deer last year, again, shortly after it was built. We currently have 3 research animals at CVERS. They're all females. I want to point out that this project is in close collaboration with Mass Wildlife. So they helped design the facility, and they're the ones that populated it with the cohort of research animals. And really on our end, what we've been busy with is just to date anyways, is just the habituation of the animals and the day-to-day sort of processes, husbandry, giving them food and water. So here's a short video of them using a bottle feeding station. But we have been thinking forward to research projects. So we're also collecting naturally attached ticks off of the deer. We did that last fall adult season. We're also doing it again now. Getting adults and nymphs off the deer. And every year we're going to get new cohorts of deer, likely in midsummer, until we reach that cap of about 15 deer in the research facility. So that gives you all an idea of what it looks like and what we've been doing to date. But of course, we built this facility with deer targeted tick control in mind. So we're looking ahead, obviously, and trying to plan out those experiments. And the first experiment we have is an anti-tick vaccine experiment. And I want to say immediately that this is really Dr. Guangzhu's research project. It's his idea. I'm just in a position to help facilitate this project and get it over the finish line, hopefully, or start it at least. But really, it's Dr. Zhu's research project. But I can hopefully give everyone just a brief overview of what it will look like. So we developed an mRNA vaccine in partnership with another lab at UPenn. And for those that are curious, it's based on the Ixodes scapularis homologue of the BM86 antigen. So some people in the audience may know that story of how there were other anti-tick vaccines targeting a different tick species, parasitizing a different host, and that was the target there, the BM86 antigen. We're using the homologue of that protein from Ixodes scapularis as the basis for our vaccine. And so this is just a real quick overview of what the experiment will look like. We're going to collect blood several times. We're going to immunize them several times. We're going to try to coincide collecting ticks with the natural tick questing activity of adults later on this fall. So this experiment is planned for later this year. And some sort of experimental outcomes that we want to measure from this experiment is we want to characterize immune response over time. So we'll collect the blood and we'll see when they seroconvert and how robust is that seroconversion, and does it differ and how long does it last, things like that. So we'll develop an ELISA for that. And we also want to measure parameters of tick fecundity or tick reproductive potential. So we'll pull off ticks and we'll look at numbers of females, numbers of engorged females, how many eggs they lay, how many of those eggs produce viable larvae, and things like that. So this project is planned again for later this year. So hopefully we can report back on that at some point later. with the results. So stay tuned on that project. Okay, so again, I'm just going to try to skip over the top of a lot of these projects. So the next one I'm going to talk about is this project, how we can use deer to study the impact of their blood on Borrelia survival. So again, we can use Seavers for this. So Seavers again is the research station that we just built, because now we have access to animals and naturally parasitizing ticks. So I'll start here. We've known for a while that deer are not reservoirs for Borrelia burgdorferi, the causative agent of Lyme disease. I'll explain what that means. We know that deer don't get infected, or at least very rarely, with Borrelia burgdorferi. And probably more importantly, for biological significance, what we find is that when larval ticks, so I'm showing here an Ixodes scapularis larval tick, when they feed, so larval ticks are, they don't have infection, they're born free of infection. When they feed, Borrelia is not being transmitted to those ticks. So deer don't get infected with Borrelia, they don't maintain infection, and they don't transmit infection to feeding ticks. So we say that they're not competent reservoirs for Borrelia. Well, what we showed in the lab a couple years ago is that if you take white-tailed deer serum and you mix it in a test tube with spirochetes and media, that it kills Borrelia burgdorferi. It's really efficient killing. It's nearly 100%. Some spirochetes maybe squeak through and survive, but really it's nearly 100% killing. And so we proposed that as a major mechanism as to this observed reservoir incompetence. What we wanted to know was whether this killing is also happening inside infected feeding ticks, so more so in a natural setting. So the question was determine whether killing Borrelia is happening inside feeding ticks. And I'm not the first person to think about this. Other studies have sort of talked about this and suggested it because they found lower prevalence of Borrelia in deer-fed ticks. So they thought, maybe deer blood is killing Borrelia, but most of those studies, as far as I can tell, were based on PCR, which detects DNA, or maybe there's a couple of, based on fluorescence microscopy early on. But what I wanted to do was really use a culture-based assay to get at this question of viability inside these feeding ticks, and also couple that with a real-time, with a PCR-based assay. And so the overview of how we did this experiment is pretty simple. I'll walk you through it. And I do want to say that this project, all this work really was done not by me, but by others in the lab. And I want to highlight those individuals as I go through the slides. So the collection of ticks was done primarily by Melina, who's a technician in the lab. The culturing was done by Nolan Stamborski, he's a master's student. And then also by Sinchana, who is an undergraduate. She did a lot of the culturing. Okay, so what we did is we collected ticks from the environment by flagging around Seavers. And then we also collected ticks off of the deer. So they would be flat ticks, meaning not fed, and engorged ticks, so some degree of feeding. And we brought them back to the lab and we mixed them up, we crushed them up in media, and we saved a small aliquot called that day 0, which we could check for presence of DNA by PCR. And then we essentially just let the cultures go and gave them the opportunity to grow spirochetes, grow Borrelia for 14 days. And we came back and we looked by direct observation, by dark field microscopy, to see if there was viable spirochetes in those cultures. So here's the results of this little study. Sample sizes are pretty small here, but I think we're getting some interesting results. So we found out of 43 flag ticks, 70% of them had Borrelia DNA from that day 0 sample, which is pretty high, but it's not unheard of in this area. And then we gave those cultures an opportunity just to grow. 63% of them produced viable living spirochetes that were observed on day 14. And then for the flat unattached ticks from deer, so we have 50 of those. So these are ticks from deer, but they haven't fed. 58% of those had evidence of Borrelia DNA, again from day 0. And then if you let the cultures go, 46% of them grew Borrelia. So then this is where it gets interesting on this bottom row, the fed-attached ticks from deer. So these are ticks that we collected from deer that had some degree of engorgement of feeding. Again, high prevalence of Borrelia burgdorferi DNA, 61%, but none of them grew spirochetes as far as we could tell on day 14. So this is telling me that the deer blood's rushing in, it's killing spirochetes in the tick. At least it's not letting them grow in culture. And if you're trying to get at this question, you really need to couple it, or at least do a culture-based assay, because you can find the DNA, and maybe those spirochetes aren't alive, aren't viable. So just to wrap that up, what we found in the lab a few years ago is that the white-tailed deer serum can kill Borrelia in vitro, right, in a test tube essentially. But then we took it one step further, and now we're demonstrating by culturing that there's pretty good evidence here that the deer blood is entering the tick and feed as the tick's feeding and killing Borrelia burgdorferi inside the feeding tick. And so we're going to we are following up on this. We're collecting more samples right now from sievers, which is also great because we can collect nymphs, which is, they're hard to collect from white-tailed deer, but we can do that with this facility. So we're collecting nymphs and adults, and we're going to, repeat this with another field season and bigger sample size to see if this trend continues. So that is project number 2. I do want to point out at this stage that these 2 projects, again, just to highlight, are coming out of or possible through the research station sievers. And then the next 4 projects I'm going to talk about are all happening or using samples that we collected during a hunter-harvested white-tailed deer surveillance program. So I'll talk about that right now. So a couple of years ago, we were chatting in the lab, and we wanted to institute a statewide standardized hunter-harvested white-tailed deer surveillance program. So a little bit more context. Every year in Massachusetts in December, it's deer hunting season in the first couple of weeks of December. And so hunters can go out and harvest white-tailed deer, and then they're required to bring them to certain locations across Massachusetts. They're called check stations, where they check them in, and Mass Wildlife collects their own data to get a good look at the population and do their own analyses. And we're using that opportunistically as a way for us to collect samples. So again, this project is also in close collaboration with Mass Wildlife. And so what we've done is for 2 years in a row, we've been to 13 sites. And you don't have to know what the letters mean, they're just the different sites. But what the colors mean is we've been there. Yellow means we've been there 2 years in a row. Purple means we were there just 1 year in 2023. And orange means we were there only in 2024. And so what we've done is that every deer that we come across, and it's not just us in the lab, we have collaborators across the state, specifically out here on the eastern side of the state where it's hard to get to. We record the information for some information for every deer, so age, sex, location, things like that. We take a blood sample in the form of a dry blood spot or a blood spot on filter paper, and then to standardize ectoparasite collections, we collect ectoparasites from the head and neck for 2 minutes, which is similar at least to how other people have done ectoparasite collections from hunter-harvested white-tailed deer. And we did this to standardize collections across space and time and among individuals. So here's all the sort of overview results of this project so far, all the samples we've collected. And I want to point out again that Melina in the lab, the technician, she's really the one that's been responsible for in processing all these samples. So, putting them away, freezing them, identifying the ectoparasites. So a ton of work coming through the door. But what we've done so far, I have it broken down by year, but we can just focus on this bottom row, is we've sampled nearly 600 white-tailed deer. We've collected nearly 600 blood samples in the form of dried blood spots. That's what DBS means, dried blood spots. We've collected over 4,000 Ixodes scapularis, or black-legged ticks. We've collected 173 Dermacin or albopictus, or these are moose ticks for people that maybe aren't familiar. As the name implies, they commonly parasitize moose, but can also be found on white-tailed deer. We found these across the state. We've collected just 37 Lone Star ticks, or Amblyomma americanum. These all came from Martha's Vineyard. And then we've collected 261 KEDs. KEDs are not ticks. KEDs are insects, and I'm going to come back to them in a different project. So a lot of work, a lot of collections so far. I want to point out that one thing we haven't found, at least on white-tailed deer, which maybe people are seeing this, is the Asian longhorn tick. But I want to point that out because this sort of surveillance is a good way to keep an eye out for those potentially invasive species that are encroaching into new territory. Okay, so then just to give everyone a quick idea of what the burden looks like, the ectoparasite burden, I just wanted to show people some preliminary data. So if you look at it over a couple years, basically 70% of the white-tailed deer had at least one ectoparasite that we could find in that timed collection window. So that means 30% couldn't find any ectoparasites. And then if you average it out, we're getting about 5 ectoparasites per deer during that collection. And then if you want to break it out by site, so these are all the site names down here. I have them, this is the burden on the y-axis. If you want to break it out by site names here. And I have this arrayed from west to east. So these are sites on the mainland. That's just one site on Martha's Vineyard. And I'm only showing check stations. I'm not showing other sites. Like we did go to butchers and things, but I'm just showing check stations here. There's a couple of patterns that maybe pop out. One is that we're maybe getting more ticks on the eastern side of the state compared to the western side of the state. If you want to look at year-to-year variability at a single site, looks like most of the time it stays the same, but there are a couple drops potentially at 2 sites. So, we haven't fully got into this data. There's a lot more work that could be done. We could break it down by, age of the deer and sex of the deer and find a resolution of where those deer are coming from. But interesting nonetheless, it's just sort of this first pass of looking at the burden. So another thing we can do, of course, is test the ticks for pathogens. So I'm just showing here 185 black-legged ticks just from 2023 that we tested for qPCR. Nolan Stamborski, again, a master's student lab, did these qPCRs. So this is just some preliminary data of pathogen prevalence in these deer-fed ticks. So this is a combination of flat and fed ticks. And we tested them for common pathogens. And what we found is that 19% of them are infected with Borrelia or have Borrelia DNA. That's quite a bit lower than what you'd expect from host-seeking ticks, flag ticks from the vegetation. But again, we think that maybe that's because that some of these are feeding on deer, and the deer blood's rushing in and killing Borrelia and degrading the DNA. And we're not the first group, again, to find this. Other people have, again, found lower prevalence of Borrelia in deer-fed ticks, and that was one of the reasons that we're thinking that, maybe the deer blood's killing Borrelia in the ticks. Then we found there's very high prevalence of anaplasma in the ticks, nearly 50%. That makes sense as well because deer are reservoirs for anaplasma, at least certain variants of anaplasma. So the ticks are feeding on deer blood's rushing in that's infected with anaplasma, and we're picking that up in the ticks. These are sort of more normal numbers, about 11% Babesia microti, 1% Borrelia miyamotoi, and then these are very, these aren't common pathogens around here. 0% for Borrelia mayonii and Ehrlichia mirus. So I just wanted to, sort of give an overview of what that looks like. But we can do a lot more work here. We can screen blood samples from 2023 and 2024 for common pathogens. We can screen the new crop of ticks for pathogens, and we keep going out and collecting more ticks to see what's there and screening them for tick-borne pathogens. So that was the overview of setting up the deer surveillance program and just, an idea of the samples that are coming in and what they look like. Now I want to talk about more specific projects that we're doing with those samples. And one of them is how we can use the samples we're collecting to investigate understudied ectoparasites. So these are ectoparasites that are on the deer. And in particular, I want to talk about KEDs. So KEDs are really interesting. Maybe a lot of people don't know too much about KEDs. I don't know. But KEDs are not ticks. They are insects. They're flies, actually. And I want to quickly go over the life cycle of a deer KED, and I'll start here. The way this works is that a winged KED, an adult winged KED, will emerge from the leaf litter, and it'll fly through the air, and it'll land on a host. And I think it's going to land primarily on, at least it's going to be looking, we think, for a white-tailed deer. That's its preferred host, as the name implies, a deer KED. And then shortly after landing on a host, a suitable host, it'll drop its wings, and it'll live the rest of its life on the deer. And it'll move around, and it'll take little blood meals, and it'll find a mate. And so it's a one-host ectoparasite. It lives its life entirely just on that one host, we think, after landing. And if it's a female, she'll give birth to essentially a fully formed pupa, as I understand it, and it'll pupate, roll off, go into leaf litter or other suitable environment, and then the winged KED will emerge later, and the life cycle will continue. And I want to point this out quickly. I'm borrowing this image or adapting this image from a great paper from some people that maybe those in the audience know, so Erica Mocktinger and Mike Scarla in Pennsylvania. We think there's 4 species of deer KEDs in the United States. So there's Lepoptina cervi up here in the Northeast. Then there's a southern species, and there's a couple species out on the western side of the United States. But they're all similar in that they're one-host ectoparasites. They primarily parasitize white-tailed deer and other cervids. But we think we just have this one species up here in the Northeast United States, and that's also what we found is just this species. So, why do we care about deer KEDs? I think historically a lot of people haven't really cared too much about deer KEDs. I think, were viewed primarily as more of a nuisance species, but they do bite people every once in a while, and of course they parasitize wildlife. They don't seem to have too much of an impact on wildlife, as far as I can tell. They may cause, increased signs of restlessness, or scratching, or irritability, or grooming. In some more extreme cases, there may even be alopecia if there's a really high burden of them. But more research is coming out, at least here in the Northeast United States, and we are finding sort of common pathogens in the KEDs. At least we're finding the DNA of those pathogens in the KEDs. So some papers have recorded Anaplasma phagocytophilum causes anaplasmosis, Burley burgdorferi causes Lyme disease, different Bartonella species that may be implicated in KED-associated dermatitis. So this is a skin condition that may be of bacterial origin through the KED bite. We don't know though for sure. But we are finding more and more pathogens in these KEDs, but we don't really know what's in them, especially in the Northeast and especially in Massachusetts. We also don't know their distribution very well in the Northeast, again, in Massachusetts before we were doing this study. So again, in one paper, I'm citing the same paper down here, deer KEDs were only recorded in 3 counties in Massachusetts, Hampshire, Worcester, and Dukes counties. So what we want to do is, using the samples, figure out where KEDs are and what's in them. We can do that with the samples that we're collecting from the deer surveillance program. And so I'm showing here just the KED collection over the last couple of years, 2023 and 2024. But basically everywhere we've looked for KEDs, we found them at almost every site. The only exception is this site up here in Northeast Massachusetts. And I think we only went there 1 year in a row, and we didn't really get a lot of deer, so that's probably why we didn't get keds. I suspect if we went back, we would eventually find them there. But we've been able to collect a bunch of keds, so about 100 in 2023, more in 2024, and to date, 261 keds across Massachusetts. So then what we can do is, of course, test them for pathogens. And before I get into that, this is a busy slide. Before I get into that, we were able to publish this data just using these results using the 2023 samples. Based on where we found the keds, we found them in 4 previously unrecorded counties, so in Berkshire, Hampden, Middlesex, and Plymouth counties. We tested them for common pathogens. I have this broken down by site. We can just skip to this bottom row. What we found is that anaplasma was in about 30% of the deer keds, Anaplasma phagocytophilum. And that was the only pathogen we detected. So we didn't find any Babesia microti, no Borrelia burgdorferi, no Borrelia nematodii, no Borrelia neonii, and no Ehrlichia murus, just Anaplasma. And so that's interesting. And like a lot of studies, it can sometimes raise more questions, about what's going on here. So again, I just have this slide, which I'll walk everyone through. That's showing, what I'm thinking, what we're thinking, and some about what's going on, and some outstanding questions. So, what I said this quickly earlier, we do know that deer are reservoirs for Anaplasma phagocytophilum, at least certain variants of Anaplasma, we think, the deer-associated variants. So anyway, so deer can be infected with Anaplasma. And what we think is going on is then the keds are feeding and they're ingesting the deer blood, and the deer are then transmitting Anaplasma to the keds. And then what we're finding is that the Anaplasma DNA in those deer-fed keds. And we're very careful in the publication to say that's really all we found. We can't guarantee anything else. We're finding the DNA of Anaplasma in these deer keds. Doesn't mean the bacteria is alive. Doesn't mean that the keds are competent vectors. But it does raise questions. So I've outlined some of those questions here. So one of them is, can wingless keds transmit Anaplasma to humans? So can this form of the ked, this variety, or this life stage, transmit Anaplasma to humans? We do know that people that come in close contact with the harvested deer, like hunters and butchers, can get keds on them that can bite them. We also don't know whether there's transovarial transmission or vertical transmission from this generation to that generation, right? So from parent to offspring. We don't know then if there's vertical transmission, if then the winged ked can transmit Anaplasma to humans. I do want to point out that we think that the Anaplasma in the keds is probably, if we're right, coming from mostly deer. Like I said previously, the deer, we think again, are infected with deer-associated Anaplasma, which we also then think is non-pathogenic to humans. So the risk may be low of a deer ked transmitting Anaplasma and then that causing disease in humans. But again, we just don't know. And then we also don't know then, can a winged ked transmit Anaplasma back to white-tailed deer? But that's an interesting question because maybe what's going on is that deer keds are somehow involved in the perpetuation or maintenance of Anaplasma in nature. And there's this cryptic white-tailed deer ked Anaplasma transmission cycle that we just don't know a lot about. So all these are questions that we don't know at this point. But one thing we're going to try to get into a little bit more is this idea of transovarial transmission or vertical transmission from parent to offspring. So again, Melina, a technician in the lab, is going to go out and collect a bunch of wing keds later this year and test them for pathogens and see what's in those, because that stage hasn't fed yet, so anything in them must have come from the generation before. Okay, so then next project is how we can use these samples to also study parasites of white-tailed deer. And, as I've been getting into this, I've been, learning more and more. And interestingly, there's a huge, or not huge, but there's a group of parasites, understudied parasites that can infect white-tailed deer. So there's a trypanosoma species, Trypanosoma cervi, that can infect white-tailed deer. There's a Plasmodium species, Plasmodium oticolii, that can infect white-tailed deer. This is a genus that causes human malaria. There's a Babesia species, Babesia oticolii, that can infect white-tailed deer. Most people are probably more familiar with Babesia microti, which is vectored by the black-legged tick and can cause disease in humans. And then there's Tularemia cervi, which is what I'm going to talk about. So Tularemia cervi is an intracellular parasite. It can infect white blood cells and red blood cells. And we think that it's vectored primarily by the Lone Star tick, or by, Amblyomma americanum. So it's in this transmission cycle where it's going back and forth between the Lone Star tick and white-tailed deer and maybe other cervids. But we think that the infections in the hosts are typically subclinical. It doesn't seem to have a huge impact on the health of the deer. If you look hard enough, you can find reports of it having negative effects on fawns, younger deer, immunocompromised animals, stressed animals maybe. But it can have a very high prevalence in wild white-tailed deer in areas where it's endemic. So there's this one paper, recent study from 2019 from Florida, where they found that basically 100% of the wild white-tailed deer had evidence of infection, 41 out of 42 of them. So what we wanted to do is look for Tularemia cervi in Massachusetts using these samples, because we have a really interesting sample set to start looking for this parasite. So we have samples from Martha's Vineyard, where there's deer, obviously, because we're getting the deer samples, the blood samples. And then on Martha's Vineyard, there's also the Lone Star tick, or Amblyomma americanum. And then we have samples from around us here in Western Massachusetts, where we have deer, of course, because we're getting the deer blood samples, but we don't have the Lone Star tick in this area. And so we wanted to test the blood samples here and the blood samples here for T. cervi. We also wanted to test the ticks. And the idea is that maybe we'll find T. cervi here, where the tick, where there's the right tick and deer, but maybe we won't find T. cervi here. And I want to point out that all this work is being done by Jackie, who is a undergraduate in the lab as part of her honors thesis. Okay, so here's the results up here in this table. And what we found is that this means white-tailed deer dry blood spots. What we found is that on the island, Martha's Vineyard, we did find a relatively high prevalence of infection, or at least we detected T. cervi DNA in 64% of the deer samples from Martha's Vineyard, 46 out of 72. And then we looked at the blood samples around us here in Western Massachusetts. We couldn't detect any Tularemia cervi DNA, so 0 out of 94. And then for the ticks, we just had a small batch of ticks, Amblyomma americanum ticks, that we also got from the deer. Only 2 of those had evidence of T. cervi infection, or about 7%. But this is preliminary data, but at least at this stage, our results do conform with the model that Amblyomma americanum are the primary vectors of T. cervi, or at least are vectors of T. cervi, because we detected them in the ticks, in Lone Star ticks. We only detected T. cervi in the deer that also had this overlapping, only where there was also Lone Star ticks present. And we didn't detect T. cervi up here where there's deer, but no T. cervi. So again, preliminary data, we need to confirm this, more testing, maybe even, different PCR, make sure we're not missing something. But interesting nonetheless, I think, at this stage. So what I want to leave people with, at least on this project, is this idea, which is that maybe we can use the presence of T. cervi in white-tailed deer as an indicator of the presence of Lone Star ticks. So here is the estimated geographic distribution of the Lone Star tick, Amblyomma americanum, in the United States. I snagged this from the CDC website. And maybe what we can do is test for T. cervi in deer at this front line or leading edge of the expanding Amblyomma americanum distribution. So in Maine and New Hampshire and Vermont, New York, and also us here in Massachusetts. And the idea is if you find T. cervi in the white-tailed deer, maybe it's a good bet that Lone Star ticks are also around in that area. So it could be a way of surveillance. Not saying don't do active surveillance, of course, but this could just be another tool in the toolkit for looking for ticks expanding their range, especially Lone Star ticks. Okay, and then the last project I want to talk about quickly is how we can use these samples to do a serosurvey of arboviruses. So deer can be used as sentinels for arbovirus activity, or they can be used to monitor arbovirus activity. And the reason for that is because deer are large mammals. They're ubiquitous across the landscape here in the Northeast United States and other places. And they're constantly getting bitten by mosquitoes and by ticks. And so then they're, of course, getting exposed to the pathogens in those mosquitoes and ticks. In this case, I'm going to talk about arboviruses. So like West Nile virus, Triple E virus, Jamestown Canyon virus, Powassan virus. They're getting exposed to those pathogens. And then they're mounting a detectable, at least, immune response in the form of antibodies. And then what's good for us, at least for researchers, is that there's a hunting season for white-tailed deer. And so hunters can go out and harvest white-tailed deer at certain times of the year and then bring them to certain locations, depending on the state. And that then you can sample them, right, for the blood to detect those antibodies. So again, using deer as sentinels for arbovirus activity, not a new idea. So I just highlighted a few papers here where they did that sort of project previously in the area. So looking for Triple E in northern New England. I think that was Maine, New Hampshire, Vermont. Serosurvey of arboviruses in white-tailed deer from New York. And then a serosurvey of Powassan virus in New England deer. I think that was like Connecticut and Vermont as well. But as far as I can tell, similar studies have not been conducted using samples collected here from Massachusetts. So that was something that we also wanted to do, was to break into this and to start this area of research as well. And so what I'm showing here are just the blood samples that we collected in 2023, because these are the samples that were sent out for testing, and I'll get there in a second. And so for us in Massachusetts, we were able to collect 283 blood samples in the form of dry blood spots. Now we also partnered with Rhode Island, and they were able to collect more blood samples from 5 locations. They were able to collect both dried blood spots and serum, 238 and 221. And these samples are great because a lot of these actually are paired samples, meaning that they were able to get a serum sample and a dry blood spot from the same animal. So in total, we got nearly 750 blood samples in 2023. And then what we did is we partnered with the New York State Department of Health at Wadsworth, specifically the Diagnostic Immunology Lab at Wadsworth. The lab director there is Dr. William Lee. And we asked them, hey, can you guys make a new assay for us, to develop a new assay, and then test our samples? And so they've been working really hard on that. And they've developed a microsphere immunofluorescence assay, or an MIA. This is using Luminex technology, which we have a little bit of experience in the lab. So this is a bead-based assay, and the samples are run at the end on the Luminex analyzer. And this assay that they've designed can detect antibodies against Powassan virus and West Nile virus, Triple E virus, Jamestown Canyon virus, and Cache Valley virus. And the little bit that I know about this assay so far is it's pretty cool. It's multiplex, so they can detect all these things simultaneously. It's high throughput because it's 96 well plate format. It doesn't take a lot of time as far as I can tell. And another thing that they're doing is they're validating the samples based on what I think you could call the gold standard, which is PRNT or plaque reduction neutralization tests. So samples that were tested by that assay, they're testing with this new assay. Anyways, so all samples were recently tested. They're not in a position to share with everybody yet, but we're working through the data now and the analysis now. But I'm hoping that we can use these results to monitor, to report back on arbovirus activity in Mass and Rhode Island, using those samples in 2023. Maybe this will be a new tool for routine monitoring, not just for us, but for other people potentially. And hopefully we can go out and get more samples and study arbovirus activity over time and space, in Massachusetts and other states as well, potentially. So with that, I'll leave it there for today. So of course, acknowledgements, NEWVEC, of course, for funding and postdoc at NEWVEC, LMZ, Laboratory of Medical Zoology, Dr. Rich, Dr. Zhu, all the undergrads and grad students, technicians, all the undergrads who are working at Sievers, helping to keep that place going day to day. That's the research station. Of course, Department of Microbiology, UMass Animal Care Services have been very helpful. New York State Department of Health, Wadsworth. Big shout out to Mass Wildlife. So really, they've been they're cooperating partners in essentially all those projects that I just talked about. They've been very helpful and are an active partner in all those projects. And then, of course, thank you to the people who helped collect the samples for the Hunter Harvested White-tailed Deer program and Rhode Island as well for, again, collecting blood samples in 2023. So with that, I'll stop and I'll take any questions.
Thank you, Pat. That was a great talk. Yeah, if anybody has questions, feel free to raise your hand or drop them in the chat. Lars, go ahead.
Yeah, thanks. Thanks, Pat. That's a really nice overview of a wide-ranging program there. So I was just wondering in terms of really commendable to be culturing ticks to make sure it's just not DNA floating around. I was just wondering in terms of how hard is it to culture live parakeets if they are there from a fully fed ticks relative to an unfed tick. So you're introducing a lot more material into the culture tube, including blood that probably has other things in it that also could grow in the media and clog it up and contaminate.
Yeah. So yeah, thanks, Lars. So that obviously is a concern. We do a surface wash on all the ticks, but I know that doesn't take care of what's happening inside the tick. We do find, anecdotally that those blood-fed ticks do have higher rates of background contamination, but we're pretty confident that we're not missing anything. Even when there is some background contamination, spirochetes can still grow. So, we find that in the flat ticks as well. No matter how well you clean them off, you can always get some background contamination. So we do find a little bit more in the blood-fed ticks, but as far as we can tell, we haven't been able to find any spirochetes in the blood-fed ticks.
Have you tried running PCR on the culture to see if there's Borrelia DNA actually in the culture? Yeah,
we've done that as well. And the thing is, though, if they're positive for Borrelia DNA on day 0, that's that DNA may be sticking around on day 14 as well. So even if you're not seeing the viable spirochetes, if you do detect DNA, it might just be there from day 0.
Yep, no, that's fair.
Yep. Yeah, Tom.
I just want to commend you for a lot of hard work there, coordinating all of that, and you opened a lot of questions that are unanswered. So that's good job for that. There was one slide that you had where, and I don't remember which one it was, maybe for the second study where you were testing, and the assessment was for Borrelia microti in the deer, but I wondered if your assay is sensitive enough to determine whether the difference between microti and otocoilii. I know later you showed that you were interested in otocoilii as well, but I was a little surprised that I don't know if it was that slide or not, but you didn't have that. You didn't have otocoilii differentiated necessarily there, and I would imagine that would be the parasite that you would be more likely to get in the ticks that were from the deer fed.
As far as I know, our qPCR assay, so this one right here that I'm showing data for, is specific for Babesia microti and shouldn't be cross-reactive with Babesia otocoilii.
I guess I'm thinking in that chart that, so you got a diminution of the Borrelia burgdorferi, and you noted that it was less than you would have expected, and that was probably a result of the specificity of the reaction between the serum and the Borrelia, and you didn't see that with anaplasma, and so you saw actually probably a slight amplification of positives than what you would expect from just ticks alone. And then with the Babesia microti, that's probably about the right rate that would be in ticks, so it wasn't having any effect. So I can appreciate that you're showing in that set of data that the blood actually does have sort of an inhibitory effect on Borrelia within the tick, which is, interesting to see, but not surprising, especially because you did such a fine job with the test tube work as well.
Yeah, Lars.
Yeah, maybe one more. First, just a comment there too, that Andreas Højgaard over here at CDC, we're using next generation sequencing for the pseudoscapularis ticks that are being tested in-house, and there is a lot of Babesia otocoilii in those ticks. It's fairly common. The other thing I was gonna for the Anaplasma phagocytophilum, is this exclusively the non-human pathogenic deer variant, or is there any of the mouse-associated human variant in there too?
So we don't know at this stage. So our PCR assay picks up Anaplasma phagocytophilum and doesn't have the ability to discern between the different variants. My guess would be that if there's a higher proportion of Anaplasma in deer-fed ticks, that Anaplasma would be, I suspect, the deer-associated variant. Same with the KEDs. I suspect that it would likely be the deer-associated variant in the deer-fed KEDs. We just don't have the capacity right now to disentangle those different variants.
Yeah, it'd be good if there could be some efforts made to do that, because I mean we started, or I should say, not me. Our group here at CDC started separating out those two variants with sequencing maybe 3-4 years ago, and it's fairly important from a public health standpoint which one you were seeing. So I think it would be, and especially for the kids,
Yeah.
I think, but for the ticks too, it'd be really nice to get a handle on which one or both are in there.
Couple more minutes if anyone has any final questions for Dr. Pearson.
Just to add from my own experience of being covered with KEDs, it's mostly that they're annoying, but every once in a while I do get pricked by one, but they hurt like crazy right from the get-go, so I don't know how long they're really going to be able to feed on a person and transmit anything from a public health risk standpoint, especially then if most of the anaplasma in them are the deer variant and not the other. But anyway, it's still a question that could be answered.
Is that something you find pretty commonly in your area of the Northeast, KEDS?
Yeah, we have lots of KEDs swarmed constantly. And I was interested that you had one of your arrows pointed, I guess it's where someone's handling the deer and then the unwinged form maybe gets on them. I don't know how that doesn't happen to me very often, but I'm not handling deer. Usually they always, I've had them drop their wings off once they acquire me. So that I find unwinged form, but I suspect that I found them not because they crawled up on me, but that they flew on me and dropped their wings off.
Yeah, I got that information sort of anecdotally from hunters who told me that the KEDs get on them. Sometimes they think they're ticks, but they're when they point them out that they're KEDs.
Yeah.
After harvesting the deer, and also butchers. When the animals are brought to butchers, the butchers have also said that the deer KEDs can get on them.
Yeah.
Tom, you used the word swarm. Did you mean that literally? Do they have a swarming behavior?
No, but if they're very abundant, they, sometimes I've had, as many as half a dozen that I have to remove at the same time. So it's like, it feels like a swarm when you're picking the stupid things off your neck.
One of the undergrads was thinking about trapping them, like making traps for them. And so I was wondering if there was a swarm that that they would swarm to.
No, I don't think that's the case, but I don't know what you would do to attract them with a trap, but that would be interesting.
Yeah, thank you all for coming. Thank you, Dr. Pearson. This was great. We'll see you all in 2 weeks on the 21st for the next work in progress.